Hello and welcome everybody to my channel Omni Bio Entrance, and today we will be discussing CUET entrance exam question paper for the subject zoology conducted in the year 2024. If you are new to my channel, please do like, share and subscribe. If you want still previous year question papers for other subjects, you can go to my channel Omni Bio Entrance. In the channel playlist C for CUET, and there you will find. Then next one, which of the following protected areas are declared tiger reserves in India? Bandipur, yes. Bhitarkanika, so that is for saltwater crocodile, not for tiger. Manas, yes, it is tiger. Sundarbans, again tiger. Kaziranga, one-horned rhinoceros, it is found in Assam. So ACD is there in option 1, so 1 is your answer. Which of the following disease in humans is caused by expanded microsatellite repeat? It is Huntington disease. It is for the CAG repeats. They are repeated more than the normal. That will result in Huntington disease. Cystic fibrosis is genetic mutation in cystic fibrosis transmembrane conductance regulator, CFTR. Okay. Then tuberculosis, you know. Then alkaptonuria, it is caused by a mutation in the HGD gene. And this gene, it is involved in the production of the enzyme homogentisate oxidase, and this homogentisate oxidase it is involved in the breakdown of amino acid tyrosine and phenylalanine. So when there is mutation, it will result in the accumulation of homogentisic acid in the body, and this is excreted in the urine, and the urine will turn dark when exposed to air. That is alkaptonuria. Okay, now next one. Microorganisms that produce antibiotics are unaffected by the same antibiotic. Okay, now what is the reason? Active pharmacophore of the bactericidal antibiotic remains bound to antibodies. Okay, I don't know whether microorganisms are producing antibodies. Of course, you can produce antibodies in microorganisms after genetic engineering, but naturally they are not producing as per my knowledge. Then antibiotics are always produced in inactive forms and get activated by the enzymatic action of target cell. Okay, that is also not correct. Microorganisms export the antibiotic using efflux pumps. Yes, that is correct. As soon as it exceeds a certain intracellular concentration. Okay, when there is accumulation of antibiotic and in the cell, the bacteria will see that it doesn't reach up to the toxic level within the cell. Okay, so what it will do, it will throw the antibiotic out through efflux pumps and thereby prevent the itself getting damaged by the antibiotic. So that's more correct. So answer is option 3. Antibiotics are made in parts and are assembled after being transported. No, that is also not correct. Then a student was asked to clone a DNA fragment flanked by BAMH1 sites into a vector cut with BGL3. The cloning was successful as these two restriction enzymes are known to create compatible sites. Based on this, what will be the restriction site configuration of these two enzymes? Indicate the point of enzymatic cleavage. So here they have given you two restriction enzymes. You have to tell which point they cut so that they will create compatible ends. Now what is this compatible end? See here, see this photograph. So here I have taken a restriction enzyme GAATTC. So this is the enzyme. It will cut here at this position G. Similarly at the bottom also it will cut here. So you will get this fragment and this fragment, that is these two fragments. Okay. Now I will be cutting the vector also with GAATTC site. Okay, at that site. Okay, this is eco R1 recognition sequence. So it will cut the vector and the gene of interest with eco R1. So here you will get two sites. Now what will happen? The vector and the DNA fragment have to ligate. So because of complementary base pairing between these sites, they will join together. That is this site GCTTAA will join with AATTCG. Here there is complementary base pairing AT, AT, TA, TA and then CG. Okay, there is complementary base pairing and these two they will join together again with the same principle of complementary base pairing. Now you are coming to your question. See this one. So here you have got the DNA fragment flanked with BAMH1. This is the BAMH1. You don't know where BAMH1 is cutting. And this is the vector which is with the BGL2. So this is the BGL2 sequence. My handwriting is very bad. I know it, but please be cooperate with me. So this is BGL3 and this is BAMH1. Now suppose we take the first option. It is cutting here. They have given a slash mark over here. So it is cutting there. So I am cutting it. That is, I get this fragment. That is GCCTAG. Now if this fragment, this portion has to ligate. Only then the DNA fragment and vector backbone can come together. Okay. So if this has to ligate to this, so this region has to be filled with complementary base pairing. So that can happen when C base pairs with G, T with A with T and G with C. That is your sequence that has to come here will be GATC. Now if you want to get GATC here, you have to digest the vector here. I mean digest the BGL3 here. So this is the BGL3 sequence AGATCT, TCT, AGA. So if it cuts here, you will get GATCT. See here, GATCT, CTAG. So this GATCT, it is complementary to CCTAG cut with BAMH1. So they both will combine and you will get a ligated product. So that means if you cut BAMH1 here and BGL2 here, you are going to get compatible ends. Okay, so that option is there in, answer is there in option 2. Then next one. During gastrulation in Synopsis frog, the blastocele becomes the gut. It is the archentron that is becoming the gut. Blastocele itself does not directly become the gut. Is filled with endodermal cells and disappears. Is filled with mesoderm and disappears. Is displaced and its original location becomes the endoderm lined cavity. That is correct. Then next one. Following statements are with reference to oogonia. Which of these is correct? Common fate of oogonia is death by apoptosis. They have asked which of these is incorrect. They have not bolded the incorrect word. You have to be careful while reading the question. Common fate of oogonia is death by apoptosis. Yes, oogonia that do not enter the meiosis, they are removed by apoptosis. So that's correct. Oogonia are able to undergo mitotic division. Yes. So oogonia, they divide mitotically to create primary oocytes. Number of oogonia grows especially during puberty. No. the number of oogonia decreases from millions at birth to 0 in case of menopause. So at puberty, the number are not growing there, they are decreasing. So 3 is the answer, which is incorrect. Oogonia are diploid cells, that is also correct. Then next one. The following is the list of steps involved in the transfer of bacterial DNA by generalized transduction. So it is involved, transduction is by help of a bacteriophage. Which option represent the correct sequence of steps? So in generalized transduction, the steps will be first infection by the phage, then there will be fragmentation of the host genomic DNA, then this host genomic DNA, it will be packaged into the phage head, then the phage will be lyse the bacterial cell and it will be released out and it will infect another bacterial host and inject the DNA into the bacterial host. This is the generalized transduction cycle. So here you see. So the first one will be injection of phage, P1 phage DNA into bacterial host. So 1 and 2 you have to see, 3 and 4 need not. So C is the first step. Then come fragmentation of host genomic DNA by phage encoded endonuclease. So 2 will be the answer. So CE. Then comes packaging of the host genomic DNA fragment into P1 phage head. That is the next step. Then release of the transducing particle into bacterial host. That is again correct. And injection of DNA of transducing particle into bacterial host. So that is the next step. So it is CEADB. Then the following is the list of steps involved in cloning a gene. Okay, now as per the sequence they have given here, the first one will be amplification of gene by PCR using Taq DNA polymerase. Here amplification of gene is your gene of interest which you want to clone into a particular plasmid or a cloning vector. So here first you have to amplify it with PCR using Taq DNA polymerase. So E is the first step. So that will be makes 3 and 4 out and 1 and 2 in. Okay, we will see after that what is happening. Then you ligate this particular amplified gene into a TA cloning vector. So TA cloning vector is a plasmid. So ED. Then they have given C, that is isolation of plasmid. Now you are, as per me, okay, as per me I am telling you, after ligation of gene into a TA cloning vector which is also a plasmid, why do you go for isolation of plasmid DNA? You already have the plasmid DNA in your hand. Okay, so C cannot be the next step. It has to be A. Because after ligation, your tendency is to clone that particular gene, that is make copies of that gene. So you transform the ligated product into bacteria and then select for the recombinants. So here it should be transformation with blue-white selection. That is, blue will be non-recombinant colonies and white will be your recombinant colonies. So you will select the recombinant colonies, grow them, then isolate plasmid from these recombinant colonies. So after ligation it has to be A, then isolation of plasmid and then confirmation of your recombinant again by RFLP analysis. But here in your option it is ED. Then isolation of plasmid, then RFLP analysis, then transformation with blue and white selection. So that is the answer as per your answer key. But as per me it is amplification, ligation, transformation, isolation of plasmid DNA and RFLP analysis. but it might be a printing mistake in the options also. Okay. Okay, then next one is match the following mammalian order with example. Perissodactyla, it is rhinoceros. A3. A3 is there in 2 and 4. Okay. Primates is lemurs. B2. So 4 is the answer. Then logomorpha is hares and the rodentia is porcupines. So 4 is the answer. Then a taxonomic unit with representative example. Brachiopods, it is lamp shells. Ctenophores, comb jellies. Nemertea, it is proboscis worms. Onychophorans, velvet worms. So answer is option 4, that is A2, B3, C1 and D4. Then which of the following statements are incorrect? Many animals with protostome development undergo spiral cleavage. One minute. Okay. Coming to the first statement, they have asked for the incorrect statement. Many animals with protostome development undergo spiral cleavage. That is correct. Flatworms exhibit a radial symmetry. No, they exhibit I think bilateral symmetry. So B is not correct. Ray finned fishes are called sarcopterygians. So, they are actually called actinopterygians, not sarcopterygians. Sarcopterygians are lobe finned fish. So C is also not correct. Lophophore is a crown of ciliated tentacles that function in feeding is correct. They have asked for the incorrect statement. So B and C is the answer. Answer is option 2. Then bacteria produce an enzyme methylase which play a role in. So bacteria utilize methyl transferase, that is methylase, for modifying its own DNA. And why does this do? This is a part of the restriction modification system by bacteria. Now once its own DNA is methylated, it will be prevented from digestion with restriction endonucleases. Okay. So its own DNA will not be digested. Whereas once the bacteria is attacked with bacteriophage and releases its DNA into the bacteria, the endonucleases will act on the phage DNA and digest it. Whereas the bacterial DNA will be saved from digestion by endonucleases because the DNA is methylated. So 1 is the answer, modification of DNA. Then next one. Dung beetles live in regions where cattle graze, quickly burying and recycling cattle droppings. Okay. Because of the dung beetles' activity, breeding habits of disease-carrying flies are reduced, and the plants upon which the cattle feed are nourished. The relationship between a dung beetle and disease-carrying flies is. So here the dung beetles are reducing the availability of dung for flies, because these flies, they have to breed, means they need dung for breeding. So it is making the life of the flies harder to survive and breed. Okay, so it is a type of competition between two different species. So it is interspecific competition. That is 4 is the answer. It is not a mutualism where both species are benefited. Commensalism where one species is benefited, other is not. Intraspecific means competition between individuals of the same species. Here it is different species, so it is interspecific competition. Then being a medical science student, it is in a very first anatomical investigation of a female body, arrange the order of appearance of the following organs of immunological importance in anterior to posterior direction in the body. So first will be the adenoids, B. Then comes your thymus, then comes spleen, and then comes Peyer's patches. So B, A, D, C, that is there in option 3. Then which of the following is not found in eosinophilic granules? Ribonucleases, cytokines, chemokines. It is histamine that is not found in eosinophilic granule. Histamine is stored in the mast cell and basophils, not in eosinophilic granules. Then next one. Correlation between the observed and predicted values of the outcome variable is known as multiple correlation coefficient. Then square root of the variance is known as standard deviation. Then next one. A protein coding gene in the mouse was mutated such that the resultant protein was non-functional. The phenotypic effect of the mutation was observed in homozygous mutant mice. Okay, that is homozygous means both the genes or both the alleles are mutated. So naturally it is going to produce non-functional protein. In heterozygous mice, so one allele is mutated whereas the other is normal, that inherited the mutation from the mother. Okay. And in heterozygous mice, that inherited the mutation from the father. So here this is a case of dominant negative mutation. So what is dominant negative mutation? It is the type of mutation where the protein will lose its function but also interferes with the function of the normal protein that is produced by the other allele. Okay, that is dominant negative mutation. Homozygous condition, both alleles are mutated. Okay, so dominant the phenotype will be of the mutated one. Whereas heterozygous condition, one allele is normal, the other allele is mutated, still it is showing the phenotype of the mutated one. Okay. So this is a case of dominant negative mutation. The gene is imprinted. First option is what they have given. The gene is imprinted. That is, it is a process that silences one copy of a gene, and this silencing of the gene is based on from which parent that gene came from, whether it is from the mother or whether it is from the father. Okay, that is gene imprinting. Then the gene possibly has more than 2 alleles. That we cannot get from the information that they have provided. The gene is X-linked. So if the gene was X-linked, then we would expect to see different phenotypic effects in males and females, because males have only 1 X chromosome whereas females have 2 X chromosome. Okay, but that is not the case here, so it is dominant negative. Then in a sucrose density gradient, what is the order of organelle sedimentation from lower to higher concentration of sucrose? So it is Golgi, then smooth endoplasmic reticulum, and then finally rough endoplasmic reticulum, because rough endoplasmic reticulum harbors ribosomes. Then next one. Select the correct statement about the membrane transport. Correct statement. Okay. Channel protein form open pores through which suitably sized molecules or ions can cross the membrane. Yes, that is correct. Carrier proteins selectively bind to specific molecules and transport them across the membrane by undergoing a conformation change. That is also correct. Transport of molecules across membranes through channel proteins and carrier proteins always involves active transport. That is not correct, because channel protein transport substances through the membranes by passively, whereas carrier protein can transport ions and molecules either passively or through secondary active transport or facilitated diffusion. So always involves active transport is not correct. Small uncharged molecules can diffuse freely through phospholipid bilayers. That is also correct. So A, B, D, that makes answer as option 3. Then capacitation is a process in which glycoprotein layer over the acrosome is dissolved. So that's it. Thank you for watching my channel. Please do like, share and subscribe if you are new to my channel. If you want still previous year question papers of CUET exam being discussed, you can go to my channel Omni Bio Entrance. In the channel playlist, search for CUET exam and there you will find. Thank you.